Hello, my name is Laura Painter, and today I'm presenting my work on the divergent evolution of the SOS1 gene across different salinity niches. The study uses comparative tools to explore how the SOS1 gene contributes to plant salt tolerance by examining different types of plants, both glycophytes and halophytes. So here is a quick overview of today's presentation. First, I'll go over the background on why plant salt tolerance matters and the role of the SOS1 gene, and then I'll explore the main objectives of my project, and after that I'll walk through the methods and show results from BLAST and expression data from GO2R, and we'll conclude with some key findings and broader implications for agriculture and plant adaptation. So overall, salt tolerance in plants, salt stress can cause major physiological problems in plants by disrupting cellular ionic balance. This can lead to osmotic stress, lower photosynthesis, and in many cases, death. Plants have evolved various responses, including ionic transport systems like SOS1. Understanding these adaptations is key to improving plant performance under stressful conditions for ecological and economical needs. For background on the organisms that I chose to study, I chose for halophytes, a salt-tolerant plant that has adapted to saline environments, such as Eutrema salsiguinium, and it utilizes special mechanism for ion transport and sequestration. And for glyphosates, which are salt-intolerant plants, I chose Arabidopsis thaliana, which has reduced capacity to manage high salinity stress. Here is our two model plants, Arabidopsis on the left and Eutrema on the right. And you'll notice that they do look pretty similar. They actually are very closely related, but they occupy very different ecological niches, which makes them ideal for a comparative genomic study. Their evolutionary proximity also helps control for unrelated sequence differences when analyzing the SOS1 gene, and so we can look for small genomic tweaks, especially in regulation, that may drive big differences in phenotype. The current genomic status of SOS1 encodes a plasma membrane sodium-hydrogen antiporter, which is crucial for exporting excess sodium from the cell. It's a key player in the salty overly sensitive, or SOS pathway, and it's controlled by the SOS2 and SOS3 gene. While it is well studied in Arabidopsis, data for halophytes such as Eutrema is still very, very limited, making it important to investigate how this gene functions in salt-tolerant species. So here is a visual representation of the SOS1's function. It's embedded in the plasma membrane and exchanges sodium for protons to help maintain ion homeostasis during salt stress. Its activation depends on SOS2 and SOS3 under high salinity conditions. So as we can see, the sodium within the cell will be swapped out for the hydrogen outside of the cell. And just like that, they've swapped, and that's how it works. So my hypothesis is that the SOS1 gene exhibits a regulatory responsiveness and selective sequence divergence in halophytes compared to glycophytes, contributing to enhanced salt tolerance. So of this, I am interested in two kind of sub-research questions. In what way do the SOS1 sequences differ? And how does the gene expression change? So for methods, I had data sources from NCBI and data sets from GEO. And for analysis tools, I used BLAST and embossed needle and GO2R for my expression analysis. So first step was a sequence analysis. So I needed to retrieve the SOS1 gene sequences for halophytes and glycophytes from NCBI. And then I aligned the sequences using BLAST, and I examined percent identity and looked at what differences were present. So here is the study term, or the search term that I looked for in NCBI, and this is the result from that search. And here is a closer look at Eutrema salsiguinium, which is our halophyte, and Arabidopsis, which is our glycophyte, non-salt tolerant. This slide shows the blast-in results using Arabidopsis SOS1 nucleotide sequence, with a filter applied to it to only show matches in Eutrema. And so we can see that I got about an 81.2% identity and over 90% query coverage, and the e-value is 0.0, indicating high significance. So all of this is above, and the alignment visualization below shows full-length coverage of the SOS1 gene with strong matching, which is the red, which shows very high scoring segments of greater than 200. And this confirms that the SOS1 is present and highly conserved in Eutrema. However, there is still meaningful divergence. These differences might reflect regulatory or adaptive substitutions, especially if concentrated in untranslated regions or regulatory introns. Next, I looked at expression analysis using GO2R. For the expression side of the study, I wanted to look at plants under salt stress versus control conditions. This was applied to both Arabidopsis and Eutrema using publicly available data sets. And the goal was to see whether SOS1 was differentially expressed in response to salt treatments. So here is the data set that I used, in which both species are in fresh and saline conditions. So this graph on the left is called a volcano plot, and it shows how gene expression changes under different conditions. In this graph, red dots are upregulated under salt, and blue dots are downregulated. And the further the dot is from the center, the stronger the reaction was towards salt. And on the right, UMAP plots are a complexity-reducing plot, helping us visualize sample-level patterns in a nonlinear format. So here, green dots are the control and purple are the treatment, and the separation indicates that there was a coordinated shift in response to salt. So overall, this slide is looking at Arabidopsis, and it shows a moderate increase in SOS1 expression under salt stress. However, we're largely seeing more down-regulated genes. This suggests that the gene is salt responsive, but may be limited in how strongly it's regulated or activated under stress. And this shows that despite having such high percent identity in the previous study, having the gene is not enough. We also need high-level, timely expression under stress. Next, we're looking at Eutrema, and here we see the SOS1 expression has increased more significantly under salt stress when compared to Arabidopsis. So we are seeing a lot more red dots on our log2 fold change. And this supports the hypothesis that halophytes not only possess the gene, but may regulate it more efficiently. To expand the analysis beyond just closely related species, I included Selenium lycopersicum, which is a tomato and a domesticated crop plant. Unlike Arabidopsis and Eutrema, tomato is more distantly related and shows low sequence similarity, as indicated by the bottom screenshot. This contrast allows us to ask a deeper question. When it comes to salt tolerance, which matters more, sequence conservation or expression regulation? By comparing a genetically distant crop to a wild halophyte, we can begin to tease apart the relative importance of structural versus regulatory adaptation. So here is the study that I used in the GEO dataset. And here are the results from using that dataset, and we're going to look at the comparison with the other two species. So here is the UMAP comparison. And here we can see Eutrema forms distinct clusters under salt stress, and this is similar in all of the other species, where we can see very distinct clusters of each treatment condition. And this indicates that all the species do seem to have some form of reaction to salt in their SOS1 gene expression levels. However, the type of expression that is seen, it differs. And this is how we can see that it differs. So in Eutrema, which is our far right one, and the one that is salt tolerant, we see an upregulation of SOS1 genes. However, in our tomato in the middle and in Arabidopsis on the left, we tend to see more downregulation. My belief is that this comes from the plant basically shutting down by not being able to upregulate the SOS1 gene enough to account for this higher stress. It is conserving energy and basically going into a survival state. And using emboss pair sequence alignment, here is the sequence alignment of both of our model species with the tomato. So on the left, we have Arabidopsis, pretty low identity at 33%, and Eutrema on the right, also pretty low at 21%. However, this is expected since these species at a baseline are not very closely related, and so we're seeing that there is a difference there. So from all these things, I was asking myself, do I have a prediction of what the tomato salt tolerance is based off of our expression findings? And it is not salt tolerant. However, its non-cultivated relatives are. And this is important because it seems that in the domestication process, it appears that we have bred out the salt tolerance gene of this tomato. So overall, our conclusion is that Arabidopsis and Eutrema share 83% SOS1 identity, but tomato shares only about 20 to 30%, emphasizing a divergent in domesticated crops. Eutrema shows both higher expression and structural differences in SOS1 under salt stress, and this supports my hypothesis that SOS1 evolved adaptively in halophytes. For broader impacts, over 1/3 of the world's irrigated land is already impacted by salinity, and projections suggest that by 2050, more than half of all cultivated land could face salinization. So this work helps us to understand how plants adapt to salt, and it has implications for lowering desalinization costs, lower land abandonment, discovering biomarkers for salinity stress adaptations, and adapting to rising sea levels by maintaining native ecosystems and restoring degraded coastal environments. For future directions, I would like to see a functional validation of SOS1 variants, an expansion of other stress-related genes in halophytes, and even expanding within halophytes to look at the SOS1 gene, as there is, like I mentioned, very limited data on that. Here are my references. Thank you.